is a X-linked dominant traits. Okay, so what happens in the X-linked dominant? It is the mutation on the X chromosome expressed in heterozygous females and hemizygous males. Okay, so even if it is present, one X is present in the male, that is hemizygous male, there will be expression, and in heterozygous female also it will have a expression. Okay, so this is the pedigree of X-linked dominant. Okay, so X-linked dominant traits do not skip the generation. There is no skipping of the generation. Affected males pass on the trait to all their daughters and none of their sons. So this will transmit to all the daughters, but none of the sons will get affected. Okay, so affected female, if heterozygous, pass the trait to about half of their sons and half of their daughters. So this will then transmit to half of their sons and half of the daughters. Okay, so this is what is the X-linked dominant trait. Okay, so the key features are X sex affected, females are affected more than the males. Generation, there is no skipping of the generation. Male to male transmission is not seen. Then father to daughter, all daughters will always be affected, and mother to child, there is a 50% risk. So both sexes, it will be affected. Okay. So sex are affected, females will be more affected than the males. So when you see that females are affected more, that means we are dealing with the X-linked dominant. Okay. So transmission rule is that affected mother will have the 50% sons and 50% daughters which are affected. If the mother is affected, 50% sons will get affected and the 50% daughter will affected. If the father is affected, all daughters will get affected, but none of the sons will get affected. So this is the X-linked dominant. Okay. So special points to mention: females, there may be milder or variable expression due to X chromosome inactivation. Some conditions, um, male lethal in utero and seen only in females. Okay. Examples are hypophosphatemic rickets, Rett syndrome, and incontinentia pigmenti. So these are X-linked dominant conditions. Okay. So just compare autosomal dominant, then autosomal recessive and X-linked dominant. Okay. Skipping of the generation is seen only in autosomal recessive. While autosomal dominant, X-linked dominant, there is no skipping of the generation. Sex ratio is equal in autosomal dominant and autosomal recessive. While X-linked dominant, you have females which are more affected than the males. Then males to male transmission is seen in autosomal dominant, autosomal recessive, but not seen in X-linked dominant. Okay, so male to male transmission not seen in X-linked dominant. Parents are affected in autosomal dominant and X-linked dominant. Parents will be carrier in autosomal recessive. Consanguinity, autosomal dominant, not concerned. Autosomal recessive, yes. If it is there, then it will have affection in the subsequent offspring, and again it is not concerned. Okay. Now coming to the X-linked recessive. Most commonly they ask X-linked recessive, autosomal recessive, or autosomal dominant. These are the three things which they ask. So in X-linked recessive, basic concept is that gene is located on the X chromosome. Okay, males are hemizygous, so only one X chromosome is present. So express disease, even mutant genes, even if mutant gene is present, it will get expressed because they will have only one X chromosome. While in females, they will usually carriers because it needs two mutant alleles to be affected. Okay, and that is rare. Okay, so this is what is the male which is getting affected, and then there is a skipping of the generation. Okay, so daughter will be, or the females will be carrier, and sons will be affected. So what are the features of such pedigree? So key pedigree features: predominantly affects the males, inherited from carrier asymptomatic mothers, there is a skipping of the generation, no continuous vertical transmission, shows crisscross inheritance, that is mother to son to granddaughter. Okay, no father to son transmission. Okay, father gives Y chromosome to the son. Okay, then carrier female clues may have affected sons and may have affected male relative. Okay, either brother or maternal uncle will get be there. So if the female is carrier, so these questions mainly in part 2 of MRCG, this particular information is very important. Even in part 1, they can ask you carrier female clues are may have affected sons. So if you have the affected sons, that means the mother will be carrier. Okay, she is not suffering, but she will be carrier. Or may have affected male relative, that is brother or maternal uncle. Okay, so if any of the mother who is phenotypically normal, but there is a affection of the brother or maternal uncle, that is suggestive that she is a carrier. Okay, then transmission pattern. Okay, what is the transmission pattern? Mother is a carrier and father is normal. Now, what is the situation here? Mother is a carrier and father is normal. So, son, 50% of the son will get affected while 50% will be normal. Daughter, 50% will be carrier and 50% will be normal. So, when carrier female marries with the normal male, there is a 50% son will get affected while 50% will be normal. 50% daughter will be carrier and 50% doctors will be normal. Okay. So if father is affected, now when you have a X-linked recessive, but the father is affected and the mother is normal. So these are the possibilities which they are asking in the examination. Okay. If father is affected and mother is normal, all sons will be normal and all daughters will be carrier. This is very simple because the father will transmit the X to both daughters. Okay. So all the daughters will be carriers and all the sons will be normal. This is the classic examination which these two are commonly asked. Okay. So important rule is that never father to son transmission is seen here. All daughters of the affected male will be carrier if mother is normal. Okay. Then trait may skip generation and affected males often linked through maternal line. Okay. So pedigree clues. Only males are affected. What suggests the X-linked recessive? Only males are getting affected. There is a skipping of the generation. There is a maternal transmission and no male-to-male transmission is suggestive of X-linked recessive. Okay. So these are the points which you should remember. Examples of X-linked recessive are hemophilia A, hemophilia B, red-green color blindness, Duchenne muscular dystrophy, that is DMD, Leishman syndrome. This also we have seen in detail. Then Menkes disease. So these are the X-linked recessive. So male patient plus maternal uncle affected. That means mother will be carrier. Think of X-linked recessive. Female usually carrier, not affected. If female is affected, consider homozygosity, which is rare, then Turner syndrome or skewed X inactivation. Okay, so X inactivation, somewhere there is a issue. Okay, so one line summary: in X-linked recessive, it will affect the males transmitted via carrier female, no father to son transmission. Okay, now male to male, if it is present, then it is not X-linked. Okay, all daughters are affected, father is affected, that is X-linked dominant. Okay, skipping of the generation, autosomal recessive, and sometimes even X-linked recessive also. There is a skipping of the generation. Equal sex distribution, it is autosomal disease, where either it is dominant or recessive, but equal sex distribution. Okay. So differentiation of X-linked recessive and autosomal recessive. How will you differentiate X-linked recessive from autosomal recessive? So X-linked recessive mainly seen in males, while there is equal affection in autosomal recessive disease. Female are rarely affected in X-linked recessive, while it is common in autosomal recessive. Male-to-male transmission is absent in X-linked, while it is present in autosomal recessive. Carrier set females it is common, while it is both sexes is seen in autosomal recessive. Generation may skip in X-linked recessive. Autosomal recessive will also skip. Okay. Then father to son, there is never transmission in X-linked recessive. It is possible in autosomal recessive. Consanguinity is less relevant, while in autosomal recessive it is strongly associated. Okay. So that is how you differentiate X-linked recessive from autosomal recessive. Okay. So only males are affected. X-linked recessive. Male to male transmission, autosomal recessive. Okay. Consanguinity present, autosomal recessive. Affected males are rare, X-linked recessive. Okay. Now coming to the Y-linked. Okay. How this Y-linked gets transmitted? That is the fifth way of transmission. Okay. Traits are transmitted via X chromosome, passed from father to all sons. Okay, father will transmit to all sons. Okay, sex affected will be only males. Transmission from father to all sons. There is no skipping of the generation. Females are never affected, and genetic location non-recombining region of Y. Okay, so if you see, only males are affected. Only males are. There is no skipping of the generation. Females are not at all affected. So this is the Y linked transmission. Okay. So clinical relevance only affect male fertility, often affect male fertility. So additional inheritance patterns are, these are non-Mendelian patterns. What we have seen are the single gene affection, Mendelian one. Apart from that, there are certain additional inheritance patterns. These are organelle or mitochondrial inheritance, which are controlled by the mitochondrial DNA. So transmission is the maternal inheritance only. So these mitochondrial DNA, which are non-Mendelian transmission, the transmission is maternal inheritance only. Okay. So transmission is from mother to all children. Father will never transmit. Okay. Both sexes will be affected, and organelles are involved are the mitochondria. Okay. So what are the limitations of pedigree analysis? Small size. So if the family size is small, ratios may not match expected. Incomplete records, the missing data if it is there. No control mating, human factors, then long generation time shows data accumulation. Incomplete penetrance, trait may skip. Then imprinting, parent of origin effect, male lethality, missing affected males, and locus heterogeneity because of the multiple genes involved. Okay. Now coming to the certain advanced concept like genomic imprinting, that is the expression depends on the parent of origin. Then another concept is a locus heterogeneity, main disease caused by the different gene. So same disease may be caused by the different gene, that is locus heterogeneity. Okay. So these are certain advanced concepts. So significance of pedigree analysis is to identify the inheritance pattern, provide genetic counseling, predict recurrent risk, and guide prenatal diagnosis. So what are the prenatal testings which are available? Ultrasound will detect the structural abnormalities. Amniocentesis will detect the genetic analysis. Chorion villus sampling will tell us about the early diagnosis, and maternal blood testing is done for the screening purposes. Okay. So postnatal or the adult testing, what you can do? You can do the newborn screening for early detection, carrier screening to identify whether it is a heterozygote status or not, late onset disease testing to predict the risk, and pharmacogenetics for personalized therapy. Okay. So what is the role in genetic counseling? It helps the couples to assess the risk of affected child, then make decision regarding pregnancy continuation and preparation of the affected child. Then important in consanguineous marriage and advanced maternal age. Okay, so we have certain questions, doctor.
Yes, sir. A pedigree shows only males affected across generation with no male to male transmission. What is the most likely inheritance? Only males affected across generation with no male to male transmission. What is the most likely inheritance? Autosomal recessive, sir. Male to male transmission.
A male to male transmission is there,
Mm,
but it
with
is
no
only males are affected across generation with no male to male transmission. Male to male is not there.
X-linked recessive. X-linked recessive.
No male to male transmission. Only males are affected.
Affected.
So,
X-linked
so it
recessive.
X-linked. So this is very common, no?
Hmm.
Only males are affected, no male to male transmission. X-linked disease. Next.
An affected father transmits a trait to all daughters, but not none of his sons. What is this pattern? X-linked dominant.
X-linked
Dominant.
dominant. Yes. It is going to be X-linked dominant because father will transmit to all daughters. That is the classical. Next.
Consanguinity is most strongly associated with which inheritance? Dominant. Autosomal recessive.
Correct. Here is going because there are chances that the alleles will come together, no? So shared alleles increase, AR expression will be increased. Next.
Which condition rules out X-linked inheritance? Skipped generation, male predominance, male to male transmission, carrier females. Which condition rules out inheritance? Skipping generation can happen if it is male predominance, male to male transmission, carriers condition rules out. Male to male transmission.
Again, male to male transmission. Okay. So that will rule out. Next.
Which proportion of offspring will be affected in X-linked dominant if father is affected? If father is affected, all daughters will be affected, sir.
Yes. All daughters will be affected. If it is a, what proportion of offspring will be affected in X-linked dominant? All daughters will be affected. Okay. Next.
Which inheritance shows father transmitting to all sons? Y-linked.
Correct. Okay. Father will transmit to all sons. Is it Y-linked? Next.
A disease skips generation and affects both sexes equally. Recessive, skip, if it is skipping generation, it is recessive and affecting
Correct.
the both sexes. Then it is autosomal. Sir,
Okay.
option B, autosomal
Yeah.
recessive.
Okay. You understood now? If it
Yes,
is
sir.
X-linked recessive, then it will not, it may skip the generation,
Skip
but it will
generation.
not affect both
Not affecting
sexes.
both sexes
Okay.
equally. Yes,
So
sir.
that is how you should differentiate. Next.
Which is a limitation of pedigree analysis? Equal sex distribution, controlled mating, incomplete penetration, short generation time. Sir, incomplete penetration.
That could be one. Yes.
Complete.
Incomplete penetrance. Next. Next.
Mitochondrial inheritance is characterized by father to son transmission, mother to all children, only males affected, skips generation. Sir, option B, mother to all children.
Yes. Because that is through maternal transmission
Mito.
only. Okay.
Hmm.
Mitochondrial disease. Next.
Which factor makes pedigree interpretation difficult? Large family size, complete records, locus heterogeneity, early reproduction. Sir, option C, locus heterogeneity.
Because sometimes multiple genes are involved.
Multiple.
Okay. So locus heterogeneity. Okay. Locus heterogeneity. Okay. So just to remember, determine the mode of inheritance. Aim is to determine the mode of inheritance, predict the risk to the offspring, aid the diagnosis plus genetic counseling. Okay. So standard pedigree symbols that also you should remember: male square, female circle, unknown sex it is a diamond, affected is shown in a shaded format, that is filled, unaffected is shown as empty, carrier is half shaded, disease is shown as slash, marriage is a horizontal line, children the vertical line downward, generations are given in Roman standards 1, 2, 3, etc. Individuals are numbered left to right. Okay. So oldest is on the left and consanguinity is shown by the double line. Important for autosomal recessive disease. Okay. Key steps in pedigree analysis: collect family data, at least 3 generations you should have. Then draw the pedigree. Identify affected individuals. Look for patterns. Every generation it is getting affected, whether it is skipping the generation, and which sex is predominantly affected. Okay. Determine inheritance and assign genotype, and then risk prediction and counseling is required. Okay. So this is what is the flow chart. So start, is the trait seen in every generation? If it is yes, likely to be dominant. If it is skipping the generation, likely to be recessive. Check sex predominance. It is more in males, it is likely to be X-linked recessive. If it is equal male and female, it is likely to be autosomal. Father to son transmission is there, then it is autosomal and not X-linked. Okay. So that is how you, so this is the same pattern which is discussed here. Okay. Just to summarize, autosomal inheritance, autosomal dominant, vertical transmission, one affected parent always mandatory, there is equal male to female affection, father to son transmission is present, genotypes will be affected will be AA, so one will be heterozygous but still affected, other will be normal, unaffected will be AA. Okay. Important rules will be if both parents affected plus child is normal, it is autosomal dominant. Okay. If both parents affected and child is normal, if both parents normal, lead to all child's normal. Okay. Then examples are Marfan syndrome, Huntington's disease, or achondroplasia. Second is autosomal recessive. Key features will be skipping of the generation. Parents usually carrier. Consanguinity is common. The affected here are shown as a small a. Carrier will be large A and small a. So unaffected parent will have affected child here. Both parents affected, all children will be affected. Okay. Then sickle cell disease, cystic fibrosis, and Tay Sachs disease. Okay. Next is X-linked inheritance. General rule is that no father to son transmission. Males are more affected. Okay. So X-linked recessive. Key feature, mostly males affected. Females are carrier. It will skip the generation. Genotype, affected male will be shown as X small a y. Female carrier will be shown as X capital A, X small a. Carrier mother will transmit to 50% of the sons will get affected. If the mother is affected, all sons will get affected. Okay. No father to son transmission is seen. Hemophilia A and hemophilia B. Then DMD, that is Duchenne muscular dystrophy, that is the disease. Then X-linked dominance. Key features are females are more commonly affected. There is no skipping of the generation. Important rule is that affected father will transmit to all daughters are affected. Affected mother, 50% children will be affected. Okay. So Rett syndrome and Alport syndrome. These are the diseases which go by this transmission. So genotype assessment strategy. For autosomal recessive, short form affected is small a. Parents must be carriers, that is capital A small a. For autosomal dominant, unaffected is a small a, while affected is usually capital A small a. For X-linked recessive, affected male is X small a y and trace via mother. Okay. So what are the clues we should catch in the question? Trait skips the generation. If it is a skipping the generation, that means autosomal recessive or X-linked recessive. Okay. Only males are affected. That means X-linked recessive. Father to son transmission, it is autosomal disease. All daughters are affected. Fathers are affected. That is X-linked dominant. Consanguinity is present. Autosomal recessive. Unaffected parents leading to affected child is a autosomal recessive. Okay. So pedigree, and there is a difference between the pedigree and the family tree. So pedigree is mainly for the medical or genetic focus, while family tree is a broad lineage. And then standard symbols are used in pedigree. There are no strict symbols in the family tree. Then disease tracking is important in pedigree analysis, while relationship tracking is important in the family tree. Okay. So limitations is that it cannot confirm X-linked, only suggest or disprove. Then small families, misleading patterns and incomplete patterns may confuse autosomal dominant. So just to summarize, most important differentiators are the generation pattern, dominant versus recessive, sex distribution, autosomal versus X-linked, father to son transmission, rules out X-linked, and consanguinity is important in autosomal recessive. So that finishes the pedigree analysis. Thank you.